Our first speaker, Sarah Suncooler, is an MD PhD student in the pathology department at the University of Rochester School of Medicine and Dentistry. She's working on her thesis in the lab of Sam McKenzie, studying TANGO2 Deficiency Disorder in a C. elegans worm model. Her clinical interests include neurogenetics and the development of improved gene replacement therapeutic strategies. She currently serves on the TANGO2 Research Foundation Early Detection Diagnostic Committee and was a recipient of the 2024 TANGO2 Research Foundation Emerging Researcher Award. When not in the lab, she can be found swing dancing, although she's standing very still right now, singing in choir, and I'm going to have to give her a hard pass on this last one because I have 3 dogs at home, but she enjoys spending time with her cat, Beatrice. So please welcome Sarah.
Good morning. All right, thank you, Dr. Satcher, for that introduction. I am, like you said, I am Sarah Suncooler, and I am an MD PhD student working with Sam McKenzie at the University of Rochester. And I'm very excited to be talking to you all today about the work that we are doing in our lab, focusing on TANGO2 Deficiency Disorder mechanism and therapeutic discovery using a C. elegans worm model. All right. Before I begin, I must disclose that I have nothing to disclose. And I want to give you a brief overview of what I'm going to be going over. So because we have so much that we can do with the mighty, mighty worm, we're going to be sharing some updates from 3 projects that we have going on in the lab, namely some mitochondrial drug screening that we've been conducting on the worms, some experiments exploring if TANGO2 might function as a heme transport protein, and finally, some metabolomics that we've also recently conducted on our worms. So, before I get started on the data, I first must tell you why the worm is so mighty. So these are microscopic worms that have been very well studied and well characterized for the last 60 to 70 years. They have a very short lifespan and high reproductive capability, so we're able to generate lots and lots of worms to do lots and lots of experiments on very quickly. Importantly, these worms also have a high metabolism and have metabolic pathways that are well conserved or are very similar between worms and between people. This means that in a disorder with a metabolic component like TANGO2 deficiency disorder, these are a good model to possibly study a genetic disorder like that. Also, crucially for all of us here interested in TANGO2, worms have 2 versions of TANGO2, which are called HRG9 and HRG10. And as you can see from these protein structures here, they are very structurally similar to the human TANGO2 protein, suggesting that they might perform a similar function in the worm. Finally, if we take these 2 HRG9 and HRG10 genes, if we mess them up and we make them unable to produce these proteins in the worms, this is called knocking them out. And because we have 2, this is a double knockout model, we find that these worms have a robust phenotype. So I'll be going into some of the phenotypic data, but generally these worms are not able to move, they don't reproduce as much, and they have symptoms of metabolic dysfunction, which gives us lots of things for us to study in the worms. All right, so the first little brief update that I want to give you all is on our mitochondrial drug screening project. So in this project, we are trying to uncover the potential other additional therapeutics for TANGO2 deficiency disorder and are trying to learn if we have any beneficial drugs, do the mechanisms of those drugs tell us something about the mechanism of the TANGO2 protein. So just for a little background on what drugs we're testing on these worms, we found this library of about 958 compounds targeting a wide variety of mitochondrial functions. So this is a little bit hard to read, but this is a summary of all of the different ways that these drugs are going to affect the mitochondria through helping with processing reactive oxygen species, which are damage that happens as the mitochondria performs oxidant respiration. to a wide variety of very specific processes. If some of you were at the mini scientific conference on Sunday, you would have heard some of the changes and damage that can occur to the mitochondria in TANGO2 deficiency disorder. So to give us a focused approach, we thought that focusing on the mitochondria would be a good idea for an initial way to test some drugs. We're also excited because many of the compounds that we are testing in this screening are already FDA approved. So there would be a high potential for clinical translation as things develop and move forward if we have any successful candidates. So a little bit about how this study was designed. So we know that our double knockout worms are particularly susceptible to oxidative stress. So when the cell performs respiration, as your body processes energy, it creates these compounds that can be damaging to the cell. This is an example of one called superoxide. And our worms tend to die when they're exposed to oxidative stress from superoxide more frequently than our normal worms. And so we are taking advantage of this by stress testing our worms with this compound called paraquat, which is a potent generator of superoxide. So it's going to force lots and lots of superoxide stress on these worms. And we are seeing if we give our worms any of these drug compounds, are they able to withstand this oxidative stress and not die? And so to test this in a high throughput automated way, we also have this fluorescent dye called Cytox Blue that's able to highlight our dead worms. So we're able to just treat our worms in these dishes and then take a picture using this fancy scanner and see which worms are glowing blue, which ones are dead, versus which ones are still able to live. And so from this, we're then quantifying how many worms are able to survive in each drug category, and then identifying compounds for further phenotyping and looking at other ways that these drugs would be beneficial to them. So just to give you a little image of what this looks like. So this is a well full of worms. Each of these little teeny tiny squiggles is a little worm. And so in this image, you can see all of the worms that are present in this little well. And then on this side, this is what one of our fluorescent images looks like. So each of these little glowing worms is sadly a worm that has passed away. So we take these images and I developed a image processing pipeline that is able to identify and automatically count worms so that I don't have to count thousands and thousands of worms by hand. Yay. And when we do this, we get lots and lots and lots of data. Which we are still in the process of working through, but this is an update about where we are after our first pass. So we went through the entire library of about 1,000 compounds, and from that, we looked at the survival benefit and selectivity. So we wanted to see which drugs help the worm survive more, and which worms help the double knockout worm specifically survive more. From that pass, we selected 88 drugs to do more replicates in since we tested each drug once. So we wanted to see if these effects held up after multiple trials. The results of this pass you can see here. So here we have mapped out along our x-axis. This is the change in survival of our N2 or normal worms. You can think N is for normal. So this is the amount that the survival was either improved over here or worsened over here. And then on our y-axis, we have our change in our double knockout survival. So what we are looking for are drugs that are going to be in this category, where they're mostly helping our double knockout survive and aren't changing our N2 worms too much one way or the other. So from this data, you can see that we did get a number of drugs that are able to help our double knockout worms survive. And so from there, we are currently conducting some other behavioral testing on these worms to see if these drugs can help outside of this oxidative stress condition. We're also working with Vandana Gupta's lab to do some cross-species analysis. So she is also working through this library in her zebrafish on different stress conditions. So we're also testing how these drugs help TANGO2 models across species and across different stress conditions. I also just want to thank our lab student, Maddie, who is either giving a very serious talk or singing karaoke in this photo. I couldn't tell you one way or the other. But she has a great poster out in the poster session, if you haven't had the chance to see it yet, that has more details about, excuse me, about our experiments. and she did some great work helping out conduct this whole drug screening process. Okay. Our next little mini highlight that I want to go over is the question of heme trafficking. Specifically, is TANGO2 a heme trafficking protein? So we know that we have gotten some questions from you guys about some papers that have come out recently, so I just wanted to address them briefly here. So there was a recent paper that proposed that TANGO2 and its homologs, or its other versions in different species, might have a role in heme trafficking. So just for a brief background, what is heme? So this is a heme molecule. So this is an iron-containing molecule that helps form the oxygen transportation system in your red blood cells. And so this previous paper worked in worms and in other species and proposed that TANGO2 homologs might be able to help take heme from areas where there is lots and lots of heme, which in humans is our mitochondria, and might be able to help transport it to areas where there is not a lot of heme. And this was a pretty significant departure than some of the other scientific consensus that has been worked on in TANGO2 in the past. So we wanted to try to look at the results of the experiments that they did in this paper. And since they did them on worms, we were able to try to see if we could replicate those results in our lab. So they conducted these experiments using these two analogs, or these two different versions of heme. So they conducted some experiments using this protein called GAP, which is heme, but with this toxic gallium molecule in the middle. So the body will take it up, and then instead of helping like heme would, it's going to poison the worms. And then for other experiments, they used this ZNMP compound, which is also shaped like heme, but just happens to fluoresce when it's absorbed into the body. And so these previous experiments had found that worms that have mutations in the two worm versions of TANGO2, which are HRG9 and HRG10, when they're exposed to this poison, they seem to have this protective benefit. So these worms are exposed to poison, but don't die. Similarly, they also found that when they're exposed to this fluorescent molecule, they see lots and lots of fluorescence inside the worms' bellies, which they don't see in normal worms, which they took to mean that these worms might have a problem absorbing heme or transporting it from areas where there's lots of heme inside their bellies as they eat it from their food and getting it into the rest of their body. So they're having this accumulation of fluorescence, and they're not suffering the effects of this poison. So we wanted to take a look at this data with our own worms. And so when we did that, when we look at this poison, we tried this poison across a bunch of different doses in our worms. And so we found that between our normal N2 and our double knockout worms here in red, we didn't see a huge difference in survival across a variety of doses. And because we also know from our work that we have done in our lab that our worms have these other defects that are happening that don't have anything to do with heme, but do affect their ability to eat their food. We also looked at this poison in this other strain of worms called EAT2, which have a mutation in their throat that prevents them from swallowing the bacteria that they eat. So they're not able to eat as much as normal worms. And so we did see that at some doses, these EAT2 worms do have a significant survival benefit when they're exposed to this poison, even though these worms don't have any problem with their heme processing, which to us suggests that these data are able to be confounded or thrown off if there is a problem with the worms eating. Similarly, when we performed our experiments with this fluorescent compound, so here we're letting the worms absorb this fluorescent molecule. When we expose them to this fluorescence, we did not see a significant difference in the amount of fluorescence in the worms. However, if we did not treat the worms with the fluorescence and we just took some pictures on them on the fluorescent channel, we do see actually a little bit of significant fluorescence in the worms, which from other studies that have been done in worms, when the worms get really stressed out, either due to not eating or due to other damage that's happening in the body, they can produce these autofluorescent gut granules. So autofluorescent just means that nothing else, no other compounds that we're giving the worm are making them glow. They just glow all by themselves. So we thought that this this autofluorescence effect could also be throwing off this data. So our takeaways from this is that we think that TANGO2, at least in the worms, is unlikely to be directly involved in heme trafficking, and that some of these abnormalities might be downstream of other symptoms or phenotypes that could be throwing off these experiments. We also recently published this paper looking at a cross-species evaluation of these versions, which we worked very closely with Dr. Satcher and Dr. Gupta to look at some of these results across a bunch of studies. And I also have to shout out another one of our students who has another poster here who also did a great job helping out with these experiments. So that's our student Olivia. So also go look at her poster if you haven't seen that and you want to hear more about heme. Okay. Finally, I want to give you a very brief teaser into some of our current ongoing works into worm metabolomics. So looking at changes that happen to worm metabolites in the absence of TANGO2. So first, just a little overview, what the heck is metabolomics? So metabolomics measures the processes of the products of these metabolic reactions. So in your cell, there are lots and lots of reactions happening at any given moment. If you took high school biology, you might remember the citric acid cycle and all of the little intermediate compounds that you might have had to memorize. And so metabolomics just is a way to study the level of these individual metabolic byproducts. And so if we then compare different strains, so we're comparing wild-type worms, our double knockout model, and some treated worms, you can measure the changes in these metabolites and see what might be changing in the worm's ability to process energy. And so when we compare our normal N2 worms and our double knockout worms, we can see some pretty big shifts in some of these sort of broad categories of metabolites. So I just want to highlight, we see big changes in carnitines, which are involved in your ability to transport fatty acids into the mitochondria. Some changes in some of the basic building blocks. So amino acids are the building blocks of proteins. These are the building blocks of DNA. And we also see some decreases in the worm's ability to process vitamins and CoA, to process oxidative stress through this thiol redox pathway, and a general decrease in the TCA or energy production cycle. And so when you do metabolomics, you get lots and lots of data back. And so one other way that we are trying to sort of sort out the forest for the trees, if you will, is we're also treating our N2s with this paraquat compound, which I mentioned back in our drug screening. And so we're inducing oxidative stress in our normal worms, and we're comparing how that looks like in our double knockout model. So why are we doing this? So we know that mitochondrial dysfunction and oxidative damage can happen in TANGO2. And so we want to be able to tell how much of the changes that we're seeing in our double knockout model are due to the actual change in the TANGO2 protein, and how much is sort of downstream of these other changes that happen that the body or the worm body might be trying to compensate for. And so we're making a comparison between the double knockout worms, which don't have functional TANGO2, and our N2 worms that have been treated with this superoxide compound to generate lots of oxidative stress. And we're trying to see what the difference is. And so this is the graphs are a little bit small and hard to read from there, but you can see that the changes in metabolism are quite similar between our stressed out and normal worms and our double knockouts. And so what we're currently in the process of doing is trying to sort through some of the things that are different between these two profiles to see what some of the specific changes to metabolism that happen in TANGO2 are. And so this is allowing us to focus on a particular area of metabolism that we are very excited about from a mechanistic perspective, that we're currently working on some mechanistic functional assays to test further. And so that's so those are just some brief highlights as to where things stand in worm world. So I wanted to thank you all for listening and all of your hard work. I want to thank the TANGO2 Research Foundation for generously funding some grants, particularly for the drug screen and for the metabolomics, and all of the people who have collaborated with us both on Team Worm and on Team Zebrafish and Team Yeast, who have really helped drive all of these projects forward. So thank you. Okay. Questions, or we keep rolling? All right, we're rolling. All right, so then I have the pleasure of introducing our next speaker. Our next presenter is Dr. Vandana Gupta. Dr. Gupta works on neuromuscular disorders with a particular focus on the molecular mechanisms underlying genetic muscle diseases, gene discovery, and therapeutic development. Her research integrates genetics, developmental biology, and translational approaches to uncover disease mechanisms and develop targeted therapies for rare neuromuscular disorders. Vandana currently serves on the TANGO2 Research Foundation's Scientific Advisory Board. Please help me welcome Dr. Vandana Gupta.
Thanks, Sarah, and thank you for the foundation for giving us this opportunity to share our work on therapies that we are developing for TANGO2 disorder. My group is based in Brigham and Women's Hospital, Boston, and we are developing therapies for a number of rare neuromuscular genetic disorder. And let me go through some of the work that we are doing in this direction. So we all know TANGO2 disorder is caused by mutation in both copies of the TANGO2 gene. And one of the biggest challenge for TANGO2 disorder is the uncertainty, unpredictability. We have the baseline symptoms of the disease that vary from patient to patient. And on top of that, a metabolic crisis can lead to a life-threatening conditions in these patients. And with a simple trigger such as dieting or infection, exercise, and many medications that we do not understand completely. So one of our goals is to understand what are all the triggers that can lead to the disease state. And secondly, can we identify therapies to help the patient to prevent and reverse the disease? A lot of work, pioneering work, has been done by Dr. Christina Miyake's group on vitamin B that has helped a lot of patients in preventing the metabolic crisis. And now we want to further add to that to develop therapies that can identify all the pathways that we can target in TANGO2 deficiency using either small molecules, using small molecule screening in zebrafish, or by gene therapy to be able to not only prevent the metabolic crisis, but also target some of the baseline symptoms and reverse them. So just to go over gene therapy, the concept is very simple. We replace the mutant copy of the TANGO2 gene with a functional copy of the TANGO2. Most of the patients with TANGO2 mutations either make no TANGO2 protein or make very small amount of TANGO2 protein. So what we propose to do is we put a functional copy of TANGO2 gene in a carrier called AAV. And AAVs are adeno-associated viruses. These are viruses that over the years, scientists have removed the harmful part and made these viruses harmless. So this whole carrier can be delivered to the TDD patients. And then this complex can reach to every cell in the body and start to make the TANGO2 protein. And the idea is that this now functional TANGO2 protein can restore the normal function in all the cells that are affected by TANGO2 deficiency. And even though this idea is fairly simple, in the past, it used to take a few decades to take a gene therapy from the lab to the clinic through a lot of trials and failures. But the progress in the last few years have taken a number of gene therapies from labs directly to the clinic because of a lot of updates in the gene therapy area, as well as faster regulatory process that can now take therapies pretty fast to the clinic. And here are some of the examples for gene therapies. N is equal to the number of patients on which those gene therapy has been tried. And a lot of therapies, like for example, this therapy has gone from the lab to the clinic within a span of 2 years. So there's a lot of hope for the gene therapy, and these therapies are targeted to all different organs, including brain, heart, which is from my institute, as well as muscle and orthopedic disorders. So now as we are developing gene therapy for TANGO2 disorder, there are a number of steps that are involved in developing a gene therapy. First, we need a robust disease model where we can test this therapy for the efficacy as well as safety before we take them to the clinic. we need to design a therapy that can reverse or restore most of the function loss that we see in a disease. And as we test this therapy, unfortunately, the mice do not have a phenotype at the baseline condition. We have tried a number of things. And what we are doing now are modeling the condition that can lead to metabolic crisis in the patient in this mice. And so that we can have a functional readout when we start to develop therapy and test therapy in this mice. As we are developing this mouse model and testing different stressor, we have also in parallel start to create the therapy vehicle that I showed you. We are using human TANGO2 genes so that any result that we get in the mice, we can directly take that to the clinic. TANGO2 deficiency is a multisystemic disease. So one of the ability the scientists have, this delivery vehicle, we can design where we can send it. So if there's a muscle disease, we can send the delivery vehicle to the muscle. If there's a brain disease, we can send the delivery vehicle to the muscle. Because TANGO2 affects a number of different organs, we are designing our delivery vehicle so that it can go ubiquitously to all the cells that are affected by TANGO2 deficiency. And our goal is very simple. Can we prevent the disease onset in babies in which the disease is caught early on and prevent metabolic crisis and associated complications? And also the patient in where the disease is already established, to what extent we can reverse the disease to the normal function of the different organs. And I'm just going to give you an example of some of the work we have done in my lab on a pediatric rare genetic disorder in developing the gene therapy. We are working on another pediatric disorder that is caused by mutations in KLHL40. So this is a recessive disorder very similar to TANGO2 deficiency. And these mice, most of these babies die within a couple of years after birth due to lack of any therapy. And we have developed these mice that show significant weakness, overall growth delays, very similar to the patients, and they die within 1 to 2 weeks after birth. So we have treated these mice with our gene therapy, and on the left are the mice that would have died without any treatment within a week or two, and on the right is the image of a mice that was treated with the gene therapy. And we have seen that we see improved survival in most of the mice that we treat with this therapy, as well as weight gain. So our goal is when we treat with gene therapy, not only to improve the survival, but also to improve the quality of life or function. And even though we have done extensive studies in this mice, I'm just going to show you one slide to show the impact of the therapy. One of these mouse is a normal wild-type mouse, and one of these mice is a mutant mice that would have died at 1 week of age. So we treated this mice right after birth, and these mice are 8 months old now. And even we cannot tell which one is which. These mice have ear tags, so we have to read them to tell which one is treated and which one is a normal sibling. So I wanted to show this data to show the reason behind developing a gene therapy for TANGO2 disease, and the hope we have that we can take the principles that we are learning from other disorders to TANGO2 disease. So just to summarize the first part of my talk, we think that developing a gene therapy for TANGO2 may help all the patients with TANGO2 mutations, because this therapy is mutation agnostic. So any patient which is not making the protein and we put the therapy back should be able to have TANGO2 protein back in their body. So now I'm going to change gears a little bit and talk about our work on small molecule-based screening in zebrafish. So while mice are great, they are mammalian, we can test therapies that we can take to the clinic. They take time to develop and grow. So we can test one therapy at a time, but if we want to screen large number of therapies, we need smaller animal models. And for that, we are using zebrafish. Zebrafish is a tiny vertebrate that you must have seen in the pet stores. The reason we use zebrafish is that most of the genome is very similar to humans. They share 70% similarity with the human. So if we have some finding in zebrafish, it's a great chance we can move that to the higher species. We get a lot of embryos from single mating, 200 to 300 embryos. So we can test a large number of drugs at the same time in this fish. And one of the most important reasons for us is that this is a larval zebrafish, about 2-3 days old. Even at that young age in zebrafish, they have all the organs similar to human. So all the organs are established. If we identify a drug that is working in, that we think is a potential therapeutic target, we can check the drug at the whole orgasm level. Because TANGO2 is expressed in so many different tissues, we can see the effect of drug in the whole organism as such versus looking at the simple organ at a time. So a few years back, we created a zebrafish model of TANGO deficiency. These are the larval zebrafish, about 5 to 6 days of age. And one of the things we noticed is that this fish was exactly similar to the control normal fish. We did not see any phenotype. They were swimming happily in the water. So we were a little surprised at first, but then we decided to look inside the muscle because muscle is 80% of the body, and a lot of patients experience rhabdomyolysis when they go into the metabolic crisis. So what we did electron microscopy and zoomed this area of the muscle about 10,000 times. So this is a single mitochondria you're looking at in a normal zebrafish. And surprisingly, when we looked at the mutants, we found about 10 to 15% of the mitochondria were defective. So this was very interesting for us that this fish are swimming happily with 10 to 15% of their mitochondria, which is defective. Mitochondria is a primary energy source in the body, and a lot of patients, when they go in the metabolic crisis, they go in the energy crisis that leads to rhabdomyolysis and other complications in these patients. So what we did next is, okay, what happens? These fish are fine when we model the clinical stressor that lead to metabolic crisis in patient in this fish. So one of the first clinical stressor we tried was fasting. These are the normal wild-type fish, and these are the mutant fish which are happily swimming now without any stressor. But when we put this fish without any food for 2 days, these are the normal fish. They are still fine. They are pretty robust model. But the mutant, they go in the metabolic crisis. They crash. So that means that, okay, no, they are able to do this normal function. Even with 10 to 15% mitochondrial defect, they are fine. But when some stressor hit, they are walking a very thin line here. When a stressor hits, they just collapse. Very similar to what we see in the patients. And then we also tested another stressor, which is exertion. This fish, in order to model the exertion phenotype, we put this fish in a very high viscosity water, something similar to honey. Now they have to invest more energy in swimming in that water. And this is a muscle structure of the fish, control fish, and two different TANGO2 mutant lines before we put them in the water. Looks overall pretty normal. But after a few hours, when we check in the muscle of this fish, this is a normal fish, wild-type fish, but the mutant fish, they start to show tears within the skeletal muscle. And some of the fish, they have the muscle structure completely collapse, go in the metabolic crisis, and eventually they never survive. So this is pretty neat that we have a model system that can model some of the things that we are seeing in this patient, because mitochondria defect is the first thing that we see in this patient, and also in the zebrafish model. And now we know that TANGO2, most of the TANGO2 is localized to mitochondria. We decided to do a small molecule screen targeting mitochondria and all the surrounding pathway like lipid oxidation, lipid formation, lipid homeostasis, as well as mitochondrial enzymes. And we started this work in collaboration with Sam McKenzie's lab and Sarah. You saw some of that work. They use oxidative stress as one of the signal in C. elegans, and we used fasting as well as exertion as two of the stressors that lead to metabolic crisis in patient in our screen. So what we did was we put few zebrafish embryos into each well and let them grow for 1 day of age. So 1 day is the age when all the organs are established, but we still do not see a mitochondrial phenotype. And then we added one drug to each of this well and gave these two different stressors and waited for 7 days. And what we found was that under fasting condition, we identified 35 compounds that prevented metabolic crisis. Exertion, we identified 5 compounds that prevented, in the overexertion condition, that prevented metabolic crisis, and 5 compounds that are common between these two. And this is very interesting finding. In Sam's group, they have also identified compounds that are different than what we have identified under a different stress condition, although 1 or 2 compounds are similar, overlap also between 2 screens. So this suggests that different stressor can lead to metabolic crisis by different mechanisms. And it is important to understand that because one therapy cannot target or prevent metabolic crisis to all the patients under all the conditions. And this is just one of the examples at the bottom. So this is a wild-type fish after fasting. These are the mutant fish that are in the metabolic crisis after fasting. Each of the dot is one zebrafish, so we have multiple fish into each group. And this is an example of one of the drugs that prevent metabolic crisis. These fish are swimming normally like the wild-type version of the fish. And similar on the right is an example from exertion, where this fish, mutant fish, have totally crashed in the absence of any of the drugs. But when we have different drugs, they show different response, and some of the drugs are more effective than the others in preventing the metabolic crisis. So at this stage, what are we doing with these drugs? We have this handful of drugs, and we are looking at, are these drugs good in preventing the metabolic crisis when the disease is not established? Or can we go further when we start to see mitochondrial defect in the zebrafish and add the drug at the later stage to see if we can, how far we can go in preventing metabolic crisis when the disease is there? Another thing is that most of the compounds actually we have identified in this process are not FDA approved. Even the library does have FDA approved compound. But even then, these compounds they are very well studied compounds. So now we know which pathways they are affecting in the cell. And we are going to those pathways and now asking the questions, are there any FDA approved drug for these pathways, for the diseases that we can use? So our goal is to test those FDA approved compounds so that we can make the translation much faster to the patients. And finally, test these drugs in cell lines and mouse model to make sure the drugs that we identify, they don't just stay till our goal is not to have a drug and publish a paper in zebrafish, but just to take the drugs to the patient by testing in different models. So just to summarize this part, we have drugs that prevent metabolic crisis associated with fasting and exertion during early disease state, and our hope is now to extend these studies further to the late disease state as well. To summarize my talk, we are developing a couple of different therapies for TANGO2 patients, and it is important to have multiple options because each therapy may not work for every single patient. So different therapies may work on patient on the basis of disease severity, kind of mutations, affected tissue, and the patient age. And in the end, we may be able to combine some of these therapies developed by my group or other groups to have the best benefit into each patient. With that, I would like to thank my lab member, especially Grace State, who is here, and she's presenting a poster. Most of the work has been done by her. So if you haven't had the opportunity to speak to her, please go and talk to her, as well as other people in my lab who have worked on the zebrafish and also supporting us in the mice work. I want to thank Sam and Sarah for the wonderful collaboration on the small molecule screening, as well as Christina's group for sharing the TANGO2 mice with us. Finally, I want to thank all the funding agencies that have supported this work. Our small molecule work has been supported by TANGO2 Research Foundation, and our gene therapy work has been supported by Muscular Dystrophy Association. Thank you. So it's my pleasure to introduce our next speaker, Dr. Augustine Lujan. Dr. Lujan is a physician scientist dedicated in advancing research in pediatric rare diseases, including conditions like TANGO2 deficiency. He holds a PhD in biological sciences and postdoctoral training in cell and molecular biology. At Hospital Saint Juan de Dieu, he works as a researcher in the neuromuscular and arrhythmia sudden death units, serving as a bridge between basic and clinical research in the field. Agu currently serves as a T2RF Research Committee member. Let's all welcome Dr. Augustine Lujan.
Good morning. Thank you for the introduction, and thank you to the TANGO2 Foundation for inviting me and giving me this opportunity to show these results. Today, I'm presenting a project that was led by Dr. Georgia Sarquilla in Spain in the Hospital San Juan de Déu. It's a pediatric hospital. And also, I want to thank her because she was very generous to share the data with me, and now I can present it here today. So this project and the results that I will show you are part of a project that was funded by the TANGO2 Research Foundation. And I have some disclosures to say. First of all, is that I'm a member of the Scientific Research Committee of the TANGO2 Foundation, but also I have to say that I was born in the tango country. No? So I'm saying that because I was born in a city at the bottom of the Andes mountain. And as you know, in Argentina, we speak Spanish. So my English is not so good. So I don't have any problem if you stop me and you say, I don't understand what you're saying, and I will try to explain it better. And some cultural data is that in Argentina, there are many religions, no? But there is one that could keep all together in one team, and this is called soccer. This is our idol, and we are living a very special moment because now there is the soccer World Cup here in the US, and this is the last World Cup of this guy. So for us, it's like the last dance of Michael Jordan or something like that. So if you can, just watch that guy. So coming back to the science. So what do we know about the cardiac manifestations in TDD? We learned a lot about the papers that were published from our colleagues, but also during these 2 days, we were talking about cardiac manifestations in TDD during the metabolic crisis. Now we understand that when the metabolic crisis appears after some triggers, the big problem is the cardiac phenotype, what is happening at the level of the heart. So there are many different kind of arrhythmias that happen in that heart. And when I say arrhythmias, it's because normal hearts has a normal beat, no? And if you have a heart that goes faster, we call it that tachyarrhythmias, and if it goes lower, we call that bradyarrhythmia. So every situation is an arrhythmia. So what we learn from our colleagues is that to stop these arrhythmias, it's necessary to add B-complex vitamins. So now we are sure that if you are under a metabolic crisis and you add B vitamins to the patients, now we can manage those arrhythmias that before was impossible. So in the group of Dr. Georgia Cerqueira-Brugada, they have a very simple question. Okay, we understand what happens in the heart during the metabolic crisis, but what happened outside those metabolic crises? What happened in between those metabolic crises? So they call it that inter-crisis period. So just now, what is happening in our kids, in their hearts in this moment. So to understand that, they start to think, okay, we can check the electrocardiograms. So now I need your participation, and I want to know who knows what is an electrocardiogram. Hands up. Yeah, most of you knows what is an electrocardiogram. But now, who knows what does it mean, an electrocardiogram? Hands up. An hour or less. Okay. Don't worry, because the first time that I see an electrocardiogram, this was my face. Because they start to talk about the heart, saying me that this is something related with the heart. And with this information, they can understand how the heart works. And for me, it was, what is that? These patterns are totally weird for me. So to understand that, I will use a very simple example. If you have a pool, you can understand this. If you don't have a pool as me, don't worry, there is a pool outside, and you can see it after this talk. So what is the main important thing of a pool? To have the water clean, no? So to have the water clean in a pool, you need two things. Bleach, that it's tons of bleach in that pool, so take care of your clothes. But the second is the movement of the water. So you need movement to oxygenize this water and to maintain it clean. And how do you make the movement of water in a pool? You need the heart. So the heart of the pool needs only three components. You have an engine with a filter here. You need pipes that will move the water from the pool to the filter and then return it again to the pool. And you need a third component that is a source of energy. So if you have these three components and you understand these three components, you could understand the heart of a human body. So the water is going from one part of the pool to the engine, filter it, and then go again to the pool. So you can alter this system, and you have two main structures that you can modify. One is the structure. If you break a pipe, something is wrong and you cannot have the movement. But the second option is to alter the electric system. So in the human heart, it's almost the same. There are many cardiologists here, so I'm a little bit afraid to do this kind of examples, but I could manage with them after. Don't worry. So let's focus on the electric system of the heart. So this is our engine or our real heart, and it's similar in the way that you have two systems instead of one, in which you move the blood from the lungs that came with the oxygen to the rest of the body. And then you have a second system that returns all the blood from the body that doesn't have too much oxygen and return it to the lungs to put the oxygen again. So to work this system, you need to pump that liquid that is inside. And to do that, there is an electrical system in our heart that starts here in something that we call sinoatrial node that make an electrical impulse, see? Okay, so this electrical impulse will allow the atrial to pump the blood that was in this compartment to the second compartment that are the ventricles. So when this electricity goes from this point to the atrium, we can observe it in the electrocardiogram as this first wave, that we call it P. Then this electricity arrives to a second node that we call atrioventricular node. And this point is very important because we'll stop a little bit the electricity, so allows that when the atrial are squeezing the blood from one compartment to the other, all the blood goes inside this compartment. So this will stop the energy and then will let pass this electricity to the second compartment to now squeeze the second compartment and move the blood to the rest of the body. So this part of the electricity, we can see it in the ECG like this big complex here. And there is a second part of the last part of the ECG, that is this T wave, that tells you that the heart is relaxed again and is ready to do again all the job. So with this information, now we can understand that in the ECG, we are seeing different parts of the heart and how the electricity is going from one part to the other, and then how the heart is relaxed again to do the job. So you can see that in the ECG, you have like V1, V2, V3, V4. So there are different patterns. This is a pattern, this is another pattern. And why they are different? Because every pattern corresponds with one receptor. So when you do this test, you put different patch, and every patch is taking information. It's a receptor of this electricity that is moving in the heart. So if you put different patch, even you can put it in the back, you can see different phases of the heart at the level of the electricity. So you can see what is happening in this heart, in the electric system of this heart from different phases. So the problem of the ECG is that I put the receptors, I receive the information, but I'm looking your heart just in that moment. So what happens if I want to see your heart for a long time? Well, now there are different options like this one, in which you can put this kind of devices, and you can record the information of that heart for 3, 5, or 7 days. But as you see here, are very useful, but sometimes for a kid could be very uncomfortable to have this, and if they want to go to the pool or take a shower, so it's not very comfortable to have it. So there is a second option or a third option that are called implantable loop records, and are very small devices that are put under the skin. And you can see here in an RRX that you have these small devices, like a small pen drive. They make a little cut and put it inside the skin. And now you can record the electrical system for up to 3 years. So you have a lot of information of that heart every second, all the days, during 3 years. So In the hospital, we have a place in which we do the remote monitoring, in which we can see all the patients all the time during the test. And as you can see here, we have more than 300 patients that we are checking every time by remote monitoring. And Dr. Sarkeja Brugada used these devices to understand what was happening in the intercrisis periods in TANGO2 patients before the B vitamins were done to all the patients, were given. So this project started with 12 patients that all of them has this ILR. And what they discovered was that without the B vitamins, 5 of them has long QTC. So now I will start to mention different kind of arrhythmias. One of them present a Brugada pattern. But something that is difficult to see in an ECG is this one, it's bradycardia. It's when the heart is going slower instead of faster. To those patients that present bradycardia, 3% pauses. Pauses is when the heart is stopped, totally stopped. So it's not a next beat in that moment. Or there is a part of the time that is stopped. So this is something very extreme, and we have a case, as you can see here, in 2020, in which one patient present a pause here and 75 events of bradyarrhythmias. Here is the pause. You can see the pulse that I show you in the ECG first, and then there is a stop of the heart. And the day after, so this was September 2, this is September 3, we observed a second pause of the heart and almost 400 bradyarrhythmias in these patients. So in this case, this study allows something was wrong. Well, these patients receive a pacemaker and now everything is okay with him. So this was very important to understand the electrical system of this patient because allow us to make a correct decision. But what happened after? What happened with the patients and the electrical system after the introduction of B-complex vitamins? Something incredible. We didn't see any kind of cardiac arrhythmias after the B vitamins. So this project was very important to individualize TANGO2 patients, to understand the importance of B-complex vitamins, and also to understand that there are some events like bradyarrhythmias that were very difficult to find in normal ECGs. So finally, I want to show that we are now looking. I talk about the electrical system, but there also could be an alteration in the structure. We are analyzing echocardiography's data, but until now there is no any difference between controls and TANGO2 patients. So at the level of the structure, seems that the heart is totally normal. Now we are contacting European centers in Italy, in UK, and in France to try to do also the European guidelines for TANGO2. We're organizing our third Spanish Tango Day. It's a Tango Day for us in which we bring all the patients with TANGO2 in Spain to the hospital, and we can make the medical checkup in 1 or 2 days, so we can see all together, and it's very important for us because in rare diseases, sometimes it's difficult to find not the differences, but stuff that are in common between patients. So when you see all together during 2 days, you start to see all these common things. And finally, we're organizing the first TANGO2 online course for the pediatricians in Spanish. And for us, it's very important because I think that we need to raise awareness to pediatricians in Latin America to find those patients that doesn't have the diagnosis yet. For me, it's totally unbelievable that in Argentina we are 40 million people and there is only one patient, and that cannot be possible. So I think it's important to raise awareness. If you don't know where are you looking, it's impossible that you find an answer or you find a diagnosis. So first of all, we need that pediatricians at least think, okay, there is a possibility called TANGO2, I will check it. Because if not, if they don't know what is TANGO2, it's impossible. And sometimes when I speak with pediatricians in Latin America, in Spain, they don't know what is TANGO2. So I think this is the principal point here. So finally, I want to thank the arrhythmia units, the neuromuscular pathology unit, all the collaborators, the TANGO2 Foundations for funding this project. And well, thank you all. I'm open for any questions. But if the question is about cardiology, please call the cardiologist. So our next speaker is Weiji Xu. Dr. Xu is a 2024 TANGO2 Research Foundation Emerging Researcher Awardee. He's an instructor in the Molecular and Human Genetics Department at Baylor College of Medicine. He did his PhD in electrophysiology at the University of Hong Kong, and then received postdoctoral training specializing in cardiology and genetics at BCM. He is pursuing academic career path in genetic cardiac arrhythmias and enjoys conducting translational research where findings in the laboratory can have an immediate impact on patients' care in the clinic. Outside the lab, he enjoys playing basketball and hanging out with his 2-year-old son. Please, Dr. Xu.
Hello, everyone. Thank you so much for the kind introduction, Lujan. So my name is Wei Xu. I am an instructor in the Molecular and Human Genetics Department at Baylor College of Medicine. It is my great pleasure to be here. And today I'll talk about some of my latest research regarding the cardiac arrhythmia and the TANGO2 Deficiency Disorder. I have nothing to disclose. First, since it's my first time here, I want to introduce a little bit more about myself and also about my scientific journey, how to get to this TANGO2 research. So I did my PhD in electrophysiology in University of Hong Kong. I started my postdoc research training in heart disease at Baylor College of Medicine in 2018. And so you might ask, what is electrophysiology and what does electrophysiologist do? I think Lu Jiang gave a really great introduction and explanation about how the electrical system works in the body and especially in the heart. So that really saved me probably like 5 minutes time of presentation. So as you all know that our human body runs on electricity. Every thought, every emotion, every heartbeat, they are all driven by electrical pulse that's generated within our body. So electrophysiology is the subject that studies these electrical signal and activity in physiology and disease. And as Lu Jiang has already illustrated, specifically for the heart, the electrical signal during each heartbeat can be captured by this specialized medical device called electrocardiogram or EKG in short. And there are many information we can extract from the EKG. For instance, normal heartbeat versus irregular heartbeat, and we can do more detailed analysis on the shape of the waveform. For example, we can measure the duration between the starting point of the spike and the end of this wave, which, as you all know, is termed as QT interval. And this QT interval is very important because prolonged duration of this interval is associated with increased risk of cardiac arrhythmias. And we'll get back to this in a bit. And I wanted to explain a little bit about how our body conducts electrical signals or electrical charge. In the physical world, electrical charge is conducted by electrical wire. How does it work in our body? So actually, our body produces a lot of these microconductors called ion channels. and these ion channel is capable of moving charged ions from one side to the others. And essentially, in every single cell of our body, these ion channel is highly present. And it is by moving these charged ions across the cell surface that our body generate electrical signal or electrical pulse. So this is like the mechanism at the microscopic level. And there are many different types of ion channels that have different preference over what kind of ions that they allow to pass through. And for instance, you have sodium channel that are specific for sodium. You have potassium channels that are specific for potassium ion. You also have calcium ion channel that are specific for calcium, and etc. And in the heart, we have found the presence of at least 4 different types of sodium channels, obviously with one that's kind of dominant, and also 4 different types of calcium channels, and I think over 15 different types of potassium channels. And as you can imagine, disruption of normal ion channel function could result in cardiac arrhythmias. For example, loss of function in the potassium channel is associated with a genetic disorder called long QT syndrome. So studying the underlying ion channel abnormalities or channelopathy, that's how we call it like in a scientific word, and is essential to understand the disease biology and also help develop targeted therapies for arrhythmia disorders. So I've been studying the cardiac arrhythmia in TANGO2 Deficiency Disorder, or TDD, since 2021. And in 2024, we published a vitamin B9 study on TDD cardiac arrhythmias. So I'll briefly summarize, in the rest of the talk, I'll briefly summarize what we have published in the past and what current research is going on in the lab. So as Lujian just mentioned, at baseline, the TDD does not affect the heart. However, metabolic crisis or metabolic stress can trigger cardiac event, or what we call cardiac stress. And during the cardiac crisis, all patients developed this long QT, as I have illustrated, and also Lujian has illustrated before. And historically, in about 75% of cases, could further evolve into cardiac arrhythmias with irregular heart beating pattern. And these arrhythmia used to be life-threatening if not managed with the supplementation of B5, but I think right now the mortality has dropped drastically, as you all heard the talk from Dr. Christina Miyake yesterday. And these arrhythmias, they don't respond well to any standard antiarrhythmic treatment. And on top of all these, we don't know what causes these arrhythmia at the microscopic level. So in our lab, we use this technique called heart in a dish to study the arrhythmia in TANGO2 Deficiency Disorder, or TDD in short. So we used blood cells from patients that had already been collected in the hospital, and we used a specialized technique to transform these cells into a specialized stem cell called iPSC. And with these iPSC cells, we can further transform them into heart muscle cells, or what we call iPSC-derived heart muscle cells. And with these cells, we can basically grow and maintain them in a dish, and so that we can study the disease in the lab. And we haven't reached the point where we could do gene therapy for human patient yet, but we could do genetic manipulation or genetic correction in the lab for research purpose. And these cells, just like the heart muscle cells in our body, they also contract and beat in the dish. So we could use this. I think it's out of bed. So we could use a specialized machine to kind of do a sort of EKG on these muscle cells in the dish as well in the lab, and get this recording and do like all the kinds of analysis that you want. So this is showing you a recording from the muscle cells that's derived from a TANGO2 patient, and as you can already appreciate, there's irregular beating patterns, and we had these cells with the genetic correction in the lab, and we found that the rhythm is completely normalized. So this kind of validate that our model really represent what we see in the patient, and also this arrhythmia, we confirm that it is indeed because of the TANGO2 deficiency. And with this powerful model for the arrhythmia phenotype, we're able to test all kinds of things that we want to test. And the first thing that we choose to test is the vitamin B from Dr. Christina Miyake's clinical observation. So there are many different types of vitamins, right? Vitamin B from B1 to B12, and I think she has some data suggest back in the day she was telling us she thought the most promising is B9. So we went ahead and put B9 on these cells, and as you can appreciate, the supplementation of B9 or folate completely reversed the arrhythmia in the TANGO2 cells. And also to kind of definitive prove that it is the action of this folate of B9 that we put into these cells, we use a drug called MTX, which is able to block the action of folate or prevent folate being metabolized in the cells, and you can see that the arrhythmia occurred again. So this is a very important data because it tells us the action of folate. It requires folate metabolism inside the cells, and and furthermore, we show that the folate action is actually pretty quick. It's able to terminate arrhythmia in about 4 hours. I believe it's even faster, but the fastest, the earliest time point we can catch is 4 hours. So I think that this was the first sort of experimental evidence to really suggest that folate or B9 is effective in prevent or terminate arrhythmia in TANGO2 deficient patients. And along with the clinical study with Dr. Christina Miyake, I think this simple but life-saving strategy has been widely implemented in the clinic. And we performed additional analysis on this durations for each waveforms, and here is a representative tracings, zoomed, enlarged tracings from TANGO deficient cells, which is shown in red. TANGO deficient cells receive B9, which is shown in blue, and cells that receive genetic correction in the lab, which is shown in black. And I put two dashed lines here at the peak of this wave, just to show you that in the TANGO deficient cells, there's a longer duration versus the duration in the cells that receive genetic correction. And this prolonged duration in the TANGO deficient cells represented the long QT feature that Lu Jian just mentioned, as we see in the patients. And what happened to the cells with B9 with folate? Surprisingly, we found that the cells receive folate, the prolonged duration is still there. It's not go back to the sort of the normal level or the baseline. So this suggests that B9 is able to terminate arrhythmias, but it does not eliminate this long QT feature in TANGO2 deficient disorder. And this got us really interested because generally speaking, cardiac arrhythmia is like a fire. So it consists of two components. One is the trigger to initiate the fire, and also substrate to be able to propagate and sustain the fire. So, what is the substrate and trigger in the TDD arrhythmias? So, as I mentioned, all patients develop the long QT feature during crisis, and long QT is actually a well recognized substrate, pro-arrhythmic substrate for arrhythmias. So we kind of know that the long QT serves as the substrate over here. What about the trigger mechanism? We don't have an answer for that yet, but what we do know is that the Foley is able to terminate arrhythmias, but it doesn't change this long QT, so it must act on this trigger mechanism for whatever it is. So to kind of further elucidate the mechanisms, we study the underlying ion channel abnormalities in both trigger mechanism and the substrate. So as I mentioned before, loss of function in the potassium channel in the heart can cause long QT. So we look at 3 long QT related ion channels, 3 major ones, which includes CKIR 2.x, HERG, KV 7.1. Don't ask me why it's named so complicated. So what we found is the activity of the CKR 2.x and HERG, I think they call it HERG appears to be normal between the TDD cells and the healthy cells. What we found is there's about 1/3 reduction in the KV 7.1 activity in the TDD cells versus the healthy cells. So this data suggests that the substrate is contributed by reduction in KV 7.1 potassium channel activities. And as far as the trigger mechanism, because the metabolic stress induced cardiac crisis, so we choose to look at ion channels that are regulated by energy metabolism. And we found that there's one potassium channel called ATP sensitive potassium channels, or KATP in short, which is able to regulate its activity based on the energy status in the cells. We found that this KATP channel is overactivated in the TANGO2 deficient cells versus the normal cells. And because overactivation of KATP has been associated with increased risk of ventricular arrhythmias in both human patients with genetic disease and also animal models previously. So this data led us to hypothesize that the overactivation of KATP may underline the trigger mechanism, and we are actively working on to test this hypothesis. And also another interesting question that we want to address is whether the anti-arrhythmic effect of Foley is through suppressing the overactivation of this KATP channel. And in the meantime, because Foley doesn't eliminate this long QT or the substrate, so we are actively working on ways to restore the KV 7.1 activity so that we can eliminate the pro-arrhythmic substrate in TANGO2 deficiency disorder. So with that, I would like to thank all the people and funding resources that has contributed to this work. On the left are the people based in Baylor. Dr. Lili Zhang is the principal investigator or the leader of the lab. And Dr. Yingchong Cao in the middle is a research assistant that has been providing cells for the entire project. Andy Yu is a very talented medical school student that helped with the analysis. And I also want to thank Dr. Christina Miyake for the collaboration over the last 5 years and providing really amazing insights from the clinical side. And obviously, I want to give special thanks to TANGO2 Research Foundation for providing fundings to support the research and also for organizing this amazing conference. Last but not least, I want to say that all these research are dedicated to all the TANGO2 warriors and families represented by all these beautiful drawings, all these beautiful pictures on this, my all-time favorite award plaque. And so thank you so much for your courage, resilience, and hope, which has really motivated our research toward better care and a brighter future. Thank you all. So our next speaker is actually my dear fellow at Baylor College of Medicine, Lauren Pico. She is a neuroscience PhD candidate at Baylor College of Medicine in Houston, Texas. Growing up alongside two brothers with a rare disorder, Lauren has always been passionate about understanding how genetic variants can disrupt brain development and activity. She conducts her thesis work in the Silito Lab, where discoveries continue to establish the cerebellum, a brain region that coordinates movement and balance, as an origin of diseases like dystonia, ataxia, and tremor. Using a mouse model, Lauren studies how loss of function alters cerebellar structure and functions to influence movement, with the goal of identifying ways to improve the lives of those with TDD. Beyond her research, Laura enjoys sharing her love of science through teaching and her volunteers with children and families affected by special needs in her community. Please welcome Lauren to the podium.
Hello, everyone. Thank you for that great introduction, Wei. Good morning, everyone. My name is Lauren. I'm a neuroscience PhD student at Baylor, and I'm so grateful to be given this opportunity to speak to you all today. This is my first TANGO2 conference, and it's been really valuable to speak to you all and learn more from you as I'm about to tell you right now. It's been so valuable to me. So today I'm going to be telling you a little bit about how a brain region called the cerebellum may have something to do with causing movement disorders in TDD. First, I have no disclosures. And I wanted to start off today by kind of introducing myself. Apart from Wei's introduction, rare disorder research is very, very deeply personal to me as both a scientist and a sister. So I grew up with two brothers with a very, very rare genetic neurodevelopmental disorder, and just growing up alongside them has always left me with a very, very deep passion for wanting to understand how genetic changes can lead to such complex, profound neurological changes in the brain. And so these questions really led me to the Silito Lab, where I now conduct research on neurological function in TDD. And TDD has a profound impact on neurological function, and I currently focus on movement disorders, particularly dystonia, which refers to sustained or intermittent muscle contractions that can sometimes be painful or result in abnormal postures, as well as ataxia, which refers to loss of coordination, and lastly, sometimes tremor, which means rhythmic shaking in parts of the body. And so many of our TANGO2 warriors have more than one of these movement challenges, which results in this really complex movement phenotype that can really significantly impact quality of life for warriors and their caregivers. And so where do we begin to look in the brain to understand why these might be happening? Because we really have fundamentally little understanding of what's going on in the brain to cause these neurological problems. And well, all of these neurological symptoms are actually well known to be caused by cerebellar dysfunction. So the cerebellum is this darkened part of the brain in the back here, and especially movement disorders. And so this is why I was really excited to kind of bring the TANGO2 project to my lab, because my lab within the Cerebellar Science Center at Baylor College of Medicine actually has specialized in looking at the cerebellum in these movement disorders for a really long time. And how have we done this? Well, we have, over the past 15 years, shown through both genetic and induced mouse models of motor disorders that changes in cerebellar structure can lead to dystonia, ataxia, and tremor, as well as changes in the functional activity of the cerebellum, kind of all loops together to also create dystonia, ataxia, and tremor. And so in creating these different mouse models of these disorders, we've actually found that there's distinct changes in firing patterns of cerebellar cells that are associated with these different movement disorders. And so basically, essentially, neurons fire electrical spikes to communicate with each other, which can be detected with an electrode, and those are the vertical lines here. And we can tell by the different spacing of the lines that spikes are happening in different patterns in these different movement disorders. And we characterize these patterns and call them signatures. And so interestingly, recreating these different patterns in healthy mice can actually in turn create ataxia, dystonia, or tremor in a healthy mouse. And so ultimately, what I'm interested in answering in my thesis project is, does cerebellar dysfunction contribute to this combined constellation of severe motor deficits faced in TDD? And if so, how? And so to approach this question, I've been working with a mouse model of TDD. And the mouse TANGO2 gene has 9 exons, just like human TANGO2. And this mouse has an exon 3 deletion to result in a loss of function of the protein. And I found that these mice exhibit episodic dystonia, ataxia, and tremor. And so I'm going to show you a few videos about how these mice, some of the features that these mice experience, and how we attribute them to dystonia, ataxia, tremor. And so this first mouse is going to exhibit an overextension of the hind limb, or an overcontraction of the hind limb, as well as a stiff curved tail, which we call a straub tail, which is indicative of dystonia. So it's going to pause here, and you'll be able to see there the overextension of the hind limb, as well as that curved, stiff curved tail as the straub tail. And this next mouse exhibits a very wide base gait, leading to almost a waddling-like phenotype, which is indicative of ataxia. You'll see it as the mouse is walking down this way. And lastly, TANGO2 mice exhibit episodic shaking, which is can be some combination of rhythmic tremor as well as these non-rhythmic jerks that these mice can experience. And so what I really wanted to focus on primarily is trying to figure out what is going on in the brain of these knockout mice. How are the cells affected and which cells are affected? And so moving on to the cerebellar cortex. So the cerebellar cortex lines the entire cerebellum, and it's made up of a diverse array of inputs that all come together to essentially shape motor output. And the heart of this computation in the cerebellar cortex are these beautiful cells lined in a row here, and they're called Purkinje cells, which are the cells that I've been recording from in these mice. So what does that mean? What does it mean to record from a neuron? And thanks to the past two speakers, Ahu and Wei, who have given a great introduction to electrophysiology. My work is lesser here to explain, but essentially we're able to record from individual neurons in an awake and behaving running mouse through electrophysiology. And how we do this is we perform surgery, so anesthetized surgery, to open a craniotomy in the skull, a small one right over where we want to record. And then after the mouse is given ample time to recover, I can insert an electrode in the cerebellum to record from any types of cells. In this example, I'm going to be talking about Purkinje cells today. And this produces a raw spike train when my electrode is right next to the cell body of the neuron. And then in a computer software, I can go in and detect out individual spikes. So each time the neuron is firing to communicate or talk to another neuron. And then from these detected spikes, what we're looking at is the interspike interval. So how far apart are these spikes spaced? And these interspike intervals can tell us things like firing rate, so how fast is the cell firing, as well as multiple measures of variation. So one of them is global spiking variation, which is just variation when you zoom out and look at the entire recording. and how variable are the spikes happening across that whole recording. And then secondly, local spiking variability. So how different is each interspike interval from the previous interspike interval that came before that? So it's a bit zoomed in. And so I wanted to actually show you an example of what this looks like in real time when I'm at the rig and I have a mouse on the wheel and I am right next to a neuron in the brain. And so there's going to be, this process, this is a voltage output that I'm listening to. And the voltage output is also split into audio outputs. So we're going to be able to hear the neuron talking. And each pop you're going to hear is actually an action potential that the neuron is firing. And they're going to happen so fast because Purkinje cells fire super, super fast in the brain. Super cool. I love what I do. It's a great technique. It's so, so fascinating. I love it. And so this mouse, this is from a TANGO2 mouse. This is a TANGO2 Purkinje cell. And so basically what I'm finding from these cells in these mice is that there's actually a change in the way Purkinje cells are firing a special type of spike called a complex spike. So Purkinje cells fire two types of spikes, simple spikes, which are on the left, which are the main buzzing you heard in that last video, as well as this complex spike, which sticks up a little bit more and has this little train of spikelets that make it kind of look like a triangle. And so if we zoom out into a trace and look at all the complex spikes, you can see that those complex spikes marked by the asterisk are happening less often than simple spikes on the bottom. So simple spikes are happening very, very fast, and then occasionally a complex spike is fired. And in this control trace, they're happening with a healthy variability. And then when we look at the TANGO2 Purkinje cell example trace, you can see that the complex spikes are spaced out more irregularly compared to the control. And so basically meaning the space is much more variable, but one cell comparing two cells is not enough. We need a lot of cells. And so looking at the cells that I've recorded so far or analyzed so far, despite the cells firing complex spikes at the same rate, they're firing the same amount of complex spikes, there is a significant difference in the variation of which they're firing these complex spikes on both of those measures of variation. The global variation zooming all the way out, as well as local variation. So zooming in from just spike to spike, there is a significant increase in variation. And so now the obvious question is, what does this mean? And the honest answer is we don't know yet, but I'm working super hard right now in the heart of all this data collection to gather a lot more cells. We need a lot more cells to really confirm this phenotype, and this is just one piece of the puzzle. And so each phenotype like this opens up many other questions, as science always does. And so to summarize, to leave you kind of on a summary of what we're working on now and the things we know so far, is that we know cerebellar firing changes can encode ataxia, dystonia, and tremor. And we know that these same features can drive disease in different genetic models. And so far in TANGO2, it looks different. And so we have maybe potential features of all three. So what I'm asking is basically, as I continue to record and analyze all of this data, how will the TANGO2 firing phenotype or signature either map onto some combination of what we know drives these movement disorders independently, or potentially create entirely new TANGO2 firing signature that is this representation of this complex movement disorder phenotype. And so although this work isn't directly testing a treatment at this stage, the first step is to really identify which cells are affected and how to be able to start to drive treatment strategies and neuromodulation therapeutics to be able to improve movement and ideally quality of life. And so with that, I would like to first thank the TANGO2 Research Foundation because this is a pretty new project and we just received our first TANGO2 grant. So that's very exciting and will help us over the next year. And I would next like to thank my lab, the Silito lab, as well as my PI who couldn't be here. He's actually in Africa right now. And I would like to thank our collaborator, Dr. Lilei Zhang. And I think, yeah, with that, I'll thank you very much. All right. And I have the great pleasure of introducing Mike Morris. Mike Morris is a dad to TANGO2 warrior Ryan and co-founder of the TANGO2 Research Foundation. His background is in technology and entrepreneurship, and Mike serves as the TANGO2 Research Foundation president and a member of the board of directors and research committee.
Thank you, Lauren. Appreciate that introduction. Welcome everyone again. Let me just get organized here. I'm going to put a couple of things down. Great to be here. I'm excited to kind of share about a project that I've been working on since I started in January, and I worked on it off and on, and Dina was pushing me to finish it up for the conference here. So Dina's here. I apologize for any anxiety that I kind of caused you, but I did get the poster printed out last Thursday, and I just finished testing it about 18 minutes ago. So here we go. So basically, it's called PubNav. TANGO2 PubNav. I just call it PubNav. First off, we'll start off with what it isn't, what it is not. So it isn't an app to teach people how to go to different pubs. So I'll pause for a second. If anyone is disappointed and they need to go use the bathroom or something else, they can do that. But what is it? The features at a glance. So I've been for about 2 years now. I would always ask Dina and you know, wouldn't it be great if we could get all these publications that are out there, and if we could gather them in one place. And when we started this, there were 2 publications. I believe now we're up to 86 publications, and so there's been an awful lot of progress. And I think it would be helpful to everyone in our community if they were able to have access to these publications. That would be the first thing. So first off, it acts as a repository, so you can get to all the papers here, and if they're what they call open access, you'll be able to access the actual article. The next thing is you can go in and you can do all kinds of filtering on the papers. You can go in by authors, topic, things like that. That's helpful. Each one of the articles will have a summary associated with it. When we do an upload into the platform, it's created automatically. We also have an option where you're able to attach other resources to it that augment the actual article. So things like podcast, infographics and things like that, and I'll get into that. This one over here is a nice little feature. I call it, just from like a branding perspective, Ask AI. So you'll be able to, at the article level or across all the articles, ask questions. And we're just using, I don't want to get too technical with it, but it's using a pattern called retrieval augmented generation, which is just a fancy way of saying everyone here has worked with, I'm sure, with GPT and Gemini and Claude. So this just takes those base models and it adds on this this extra data, which happens to be these articles and these publications. And so when you ask a question, it's just going to focus just on that article or just on our repository. And then this other piece at the end, I just added on about 2 weeks ago. I think this is, as I was thinking about it, this is a way for all of us to all come together as a community, parents, families, and researchers, and kind of have conversations around these publications. So with that, I'm going to get into a demo. So I have to get into the Zoom webinar here. And for those of you in our studio audience, this is the first time we've attempted this here on the stage live. So right. And let me do my share screen. Bear with me for just a second. All right. All right. It's not coming up, Chris. Yeah. Hold on a sec. Yeah. It looks like I'm sharing the right screen. There it is right there. Okay. So hopefully it doesn't have that kind of a lag when I'm actually going through the application. All right, perfect. This is going to work out well. So I'm going to get into the app. TANGO2.pubnab.app. It's a pretty little splash screen. So when you get in, it's going to have all the publications. Like I said, at this point, we've got 82 of them up there. Dina, I think we actually have 86 of them. We'll get those uploaded after the conference. And hopefully everyone who was up here prior to me, they were all talking about these great projects that they're working on, and all of those at one point or another will be published, and they'll end up in this repository here. So I can come into here, I can just do basic searching. So if I wanted to look at all of the papers affiliated with Dr. Sasher, I can come, oops, I have to spell his name correct. There we go. You can do that. If you want to look at, or if you want to go and buy papers that have in the abstract or the title, cardiac, you can do that. So there's some nice features there, or you can come in if you just want to go by the primary author, journal, things like that. This is an interesting one. I think it's a good, so I'm going to go back to, I don't know what that one is back in 06. I'm going to go back to here. So this is what we had in 2016. And then you can go up the line, and it's a nice little progression. Once we get to like 2020, we got a little bit of traction, and I think it was like 2022. I mean, there's a whole bunch of papers that came out in 2023. So I'm going to get into one of the articles. So let's get into this one right here. First one here. So when you get in, there's that abstract that I was talking about. Fairly easy to read. Here's the abstract. And then inside of it here, it's got the authors. We have a summary, which I guess is an abstract of the abstract, but detail. Go to the bottom. And then it has the affiliation with the university. But then you can also click over here and you can get into the source document. And you can look at that. Let me just get this out of the way on my own screen. So you can bump that up if you want. You can look at the pages, download, and things like that. Once again, if you'll have access to this if it's open access, download. If it isn't open access, we still kind of have to figure that out. I'll talk to our lawyers. Haven't done that yet, but And so that's there. And then here's an interesting one. I love this one. So I'm going to get into another example. I talked about those resources, the other resources. So I'm going to get into a paper here by Dr. Lujan. And, oops. Let me see. I thought that was it. Oh, here it is. I'm filtered here. Let me do a clear on the filters, and let me go in by a goo. All right, so here we are. So here's one of his papers he was involved with. Once again, you can go to the abstract, you can go to the actual source. But then, just as a sample, I've done an upload over here of a couple of resources. So these are just to kind of demonstrate the potential capabilities of what we can all do as a community. I took the article, the base article, and I uploaded it to a platform called Notebook LM, and it's a great AI platform by Google, and it can create all kinds of additional content around that article. It can create infographics, podcasts, slide decks. So you can just visualize it as a parent. You know, maybe that article for you is a little bit dense, but you can come in here and maybe you look at this little infographic and it starts to make sense to you. Here's one. I don't want to, I think if I click on the podcast, I don't think you'll be able to hear it. Let me see if I turn up my volume.
Relies completely on a very
It's going to be feedback. Yeah, that's not going to work well, but let me turn off my volume. So what I did was uploaded it there. I asked it to put together a podcast. And if you would listen to this podcast, it's like an NPR level podcast. You have two hosts kind of going back and forth. And I'll listen to them when I'm on the lawnmower, So honestly, I'll just put them on, and they're easy to understand. And here's another example. I just asked it to put together in the article a slide deck. Let me just kind of, bring this down a bump, right? But this is, you can picture as a parent or a person, or I would imagine even as a researcher. If you don't want to read that entire 16 pages, here's a little deck that was put together just to get more information on the article and things like that. So I thought that was pretty cool. All right, now I'm going to log into the application, because if I come over here, if I go to this Ask AI tab, I'm not able to actually ask any questions. But if I log in, I'll be able to do that. Now, when I log in, it'll add on this additional home page. It's going to have over here on the left there, as we kind of add articles into the library, add other resources into the library, it'll kind of list those over there. But then on the right over there, it's I call it jump back in. So this is previous activity that I was kind of playing with. So I was looking at some articles, I was asking some questions, some Ask AI. So you'll be able to kind of easily get back into your activity once you get in. But I'm going to get into an article here. So I'm going to go to publications. And earlier you heard Sarah, she was talking about that paper on heme. So here we go. So I'm going to open up that paper. Abstract, we have our source document, but here I'm going to go to Ask AI. And let me just go over here. I have a question that I kind of had pre-filled out. And I'm going to say, let me go here, put that in here. What methods were used to challenge the role of heme, and what were the results? But I, as a parent, I can also say, keep it simple. Keep it simple. It's been 40 years since high school biology. All right. I lied, it's been 42. All right. So we're gonna let that run. It'll take a little bit of time here, but I think that this is interesting to be able to go into this article and just kind of ask more pointed questions. And the nice thing with this, with them being uploaded into this, what they call a vector database, and using this retrieval, retrieval augmented generation, it's just going to focus on this content, and so you hopefully aren't going to get the hallucinations that you might get with the LLMs in general. So how they tested the heme idea, and if any of this is incorrect, Sarah, please point it out, or misrepresented. But just so here I am as a parent. I took biology 42 years ago, but I can get the overall gist of this, right? And you can even ask it like more pointed questions about an aspect of the paper. So that's pretty cool. I thought that was cool. But the bigger piece is being able to kind of leverage AI to kind of ask questions across the whole corpus of published data, right? And I think this is something that can be useful to both parents and the researchers. We have 86 papers now. Hopefully in 2 years, we have 186 or whatever. So as that grows, I think it is going to become more useful. So I'm going to go here up to the top. So this will, instead of asking it at the article level, I'm going to ask it at the, and I have one of those. So give me an overview of all of the projects that have used iPSC cells, including methods and results. And I'm just, I'm doing this for the first time. I don't know what it's going to come back with. We'll see. So it's kind of interesting how there are times when it'll come back in 12 seconds, 12 to 15 seconds. And another time, I guess it has to do deeper thinking when has to look at everything, but be curious what it comes back with. So I think, even as a researcher, I think you can kind of see the value in this if you wanted to quickly figure out, what are the animal models that have been used in the past, and in what experiments you can easily get those kinds of things. So I'm just going to go down here for a sec, and we aren't going to go over all this, but I just want to I'll give everyone an overview of, just kind of a overview of its capabilities. So this goes on for a while. So you can imagine if you had to do this manually, right? So people are really good at kind of finding obscure patterns, things that maybe there wasn't like an obvious connection to, or and things like that. And but these LLMs, they can just look at vast amounts of data quickly, and so my hope is we're able to bring together the people aspect and the AI aspect. All right, let me see what else I had here. All right, so that's that. So when you ask this, you can always go back to it. It's going to bring up your chat history here. All this is private, right? So you can come up here, you can ask all your questions. This isn't, you're the only one who kind of has access to those answers and those questions. But what I did was, a couple of weeks ago, I wanted to kind of figure out how we kind of bring this community together. So for each of these articles, or these the overall publications, these AI chats, and for the other resources, you can actually create a conversation out of them. So let's say for this one over here that I just had, if I wanted to kind of put this out to the broader community and ask other parents, other researchers, I can create a conversation out of that, right? I'm just gonna, what do people think about this, right? And oops, there we go. I'm just gonna put something stupid in there. Start conversation. And so now what's gonna happen is it ends up over here in the conversations, right? So I can go to conversations. There it is. I'm going to come into the publications. And the other next thing that I kind of have to build in that isn't built in at the moment is I'd like to, I know, every couple of weeks for people who have an account in the system, you'll get a digest. Here's what's going on. Would you like to kind of participate in the conversation? These are the articles that have just been recently uploaded. These are the other resources that have been uploaded for this article. So build it up over time. And that's kind of my hope for it. On a broader basis, I'm just inviting everyone to use this, engage in it. I think it has a lot of potential to kind of bring our community together. What I'm hoping for, and I don't know if this will happen, it's difficult to predict how these things go, but if we can get this community engaged with that, and if we can get people to use it, and if we can demonstrate value, I'd love to kind of take this and kind of bring it to other organizations, right? There's a bunch of rare disease organizations out there. Use that as a revenue stream for the foundation. And that's my message is use it, improve it, and we'll be able to perhaps do that. Okay, so here we are. Yep. So there it is. You can access it now. You have a QR code, or you can just type it into your browser. Works well on the phone as well. Use it, and I kind of built it. I've done some pretty decent testing on it, but I don't have a team to help me test it anymore, like I used to have when I had my other company. So be gentle with me, but be honest. So, right? That's it.